and slight negative on oxygen. Now, what is the significance of this? In a beaker of water, there are many hydrogen, many water molecules, and you are right between the hydrogen and the oxygen, because the oxygen has a slight negative charge on it, because the hydrogen has a slight positive charge on it. Opposites attract, right? Opposites will attract each other. As a result, we have the formation of a different kind of bond. Right? That is a covalent bond. This is what we call a hydrogen. In the hydrogen bond, you'll be talking about that a little bit today. You'll be reading about it a little bit in your lab today. A hydrogen bond is a bond that exists between two molecules, not within the molecule. There are exceptions to it. If you have a big giant molecule like a protein, there may be hydrogen bond holding individual parts of the molecule together. But generally speaking, when we talk about hydrogen bonds, we're talking about attraction between two molecules.
And the molecules are always going to be hydrogen.
Most of the time, it will involve hydrogen, which is what we call the hydrogen bond. Not necessarily all the time. Sometimes carbon has the ability to form a hydrogen bond.
So attraction between
two molecules that are both polar, and it's attraction between the slight positive charge of one polar molecule to the slight negative charge of another polar molecule. But I believe Dr. Maggie is going to cover that in much greater detail in the lecture, so wait for him to go through that. But for now, we need to know a little bit about a hydrogen bond, because when we talk about your DNA molecule, when we talk about how the two strands of DNA molecule are held together, and you will talk about that a little bit in your lab today, and you actually get to draw the diagram showing where the hydrogen bonds are. At that point, you want to know that this is how they're coming together. The two DNA strands are not held together by covalent bond. They're held together only by hydrogen bond, which makes it relatively easy for us to separate the two strands to access information. And that's why those are important. All right. So any questions about covalent bond, hydrogen bond? Ionic bond. Do you guys know about ionic bond? Right. Sometimes, if you have, say, chlorine, for instance, chlorine atom is in column number 7. Because it is in column number 7, you'll find that chlorine has exactly 7 electrons on its outermost shell. Sodium is in column number 1, means it has exactly 1 electron on its outermost shell. Right? In order for chlorine to become energetically stable, right, the easiest way to do it is to acquire 1 electron. The easiest way for sodium to become energetically stable is to get rid of that electron. And when that happens, because now sodium is missing 1 electron, it's given up 1 electron. Initially, the number of protons equals the number of electrons, charge equals to 0. Now you have 1 less electron than you do proton, so sodium acquires a positive charge. Notice I did not put the delta there, because this is 1 full positive charge, 1 entire positive charge associated with the missing electron. Chlorine, it picks up an extra electron, giving it a negative charge. Because this is positive, this is negative, there's attraction force between those two. That is where your ionic bond comes in. Right, so the difference.
They're combining, right?
They are coming together because of the positive and negative charges. So the forces holding them together is associated with magnetism in a sense. And the difference between a ionic bond and a covalent bond is actually a little bit stronger, quite a bit stronger than an ionic bond. And ionic bond, when you take the ions and dissolve it in water, it comes apart very easily. Question?
Could we just use like a non-metal and a non-metal equal the
Non-metal, pretty much that. And in biology, the only ionic bond that we really look at quite a bit is those in the 7th column. Right? Those transitional elements that I skip over, those things that I say I have no knowledge about, most of them are the metals, they're not organic. So most of those transitional elements in that middle section right there, that is where you find most of the ionic bonds. They are very, very important. Now, and the reason this thing dissolves in water very easily. Sorry, go ahead.
So just attached
They are not
by
sharing
the
electrons. Now the electrons have gone completely over.
So they're just attached by the charge.
They're just attached by the charge. Which is why when you dissolve them in water, and of course your water is separated too. So in this solid state, if you take NaNCl, which is basically a table salt, you take your table salt, take it out of your salt shaker, you look at it, it's a nice little crystal. What is happening there is that you have a sodium ion next to chlorine, next to a sodium, next to a chlorine, next to a sodium, next to a chlorine. And by arranging the ions that way, they hold each other together, which is why you have the crystallized form of salt. The moment you put it in water, the slight negative charge on the oxygen will surround itself around the positive charge of the sodium, thus separating it from the chlorine. Now the negative charge on the chlorine will be balanced off by all these hydrogens associated with the water molecule, and this is what we call a hydration shell. This water molecule basically going in and forming a shell around each of those ions, thus separating them visibly. And that causes the salt to actually dissolve in water. And that is the importance of polar covalent bonds and how it affects ionic compounds. Alright? So when you're doing your lab today, when you're asked to draw a covalent bond, use a straight line to link up between two atoms, and that is going to be a covalent bond. Right? So the first circle? CO2. Oh, CO2. So CO2 is the one actually. We haven't talked about double bonds or triple bonds. Carbon has exactly 4 electrons on the outside. Alright. In order for carbon to become energetically stable, it needs 4 more. One way to do it is to pick up 4 hydrogen to share 4 electrons with them. You can also allow carbon to share electrons with oxygen. And you remember oxygen, because it is in column number 6, yeah. 1, 2, 3, 4, 5, 6. Oxygen is a little bit too big. Right. So if you look at just the oxygen, it has 1, 2, 3, 4, 5, 6, 7, 8. This oxygen is stable. This oxygen is stable. This carbon, if you look at the carbon alone, 1, 2, 3, 4, 5, 6, 7, 8, this carbon is also stable. So this is actually a stable structure. So we're looking at now, because I say each line, each covalent bond represents 42 electrons, because there are 4 electrons holding this carbon to this oxygen, we will actually drop 2 ones down. And this is the structural formula of carbon dioxide, CO2. Right? So when you want to do a double bond, it is possible for elements to form double bond. It is possible for carbon to form single bond with another carbon. Now this is not complete, so we can complete it by adding hydrogens to it. Or we can have carbon forming double bonds with another carbon. And if you do that, then you do not need as many hydrogens going on. And in lab today, you will actually be what? Triple bonds, yeah. Yeah, and in the lab today, you will actually be constructing fatty acid molecules, fat molecules. And the difference between a saturated fat molecule and an unsaturated fat molecule, you kind of look at whether or not it has double bonds together. Right? This is considered unsaturated, and of course the fatty acid has a lot more molecules associated with that. This is considered unsaturated because it is not completely filled with hydrogen. This is saturated because it's saturated with hydrogen. You cannot add any more to that. Right? And then of course we talk about hydrogenated fats, trans fat. What is happening is you add to convert the double bonds into single bond, and it raises a whole bunch of health issues. All right? Questions? Questions? Carbon and oxygen. This one, the bond will, because the oxygen is here, it will drag more electrons to it. But this molecule is considered non-polar because there is no separation. Two oxygens on that and the both sides. Symmetrical molecules generally will be non-polar because the sharing of the electrons will be equal on this side as it is on that side. Yeah. Water.
Meaning what? Meaning two, one on the other.
Say again?
Symmetrical means there's two on each side.
Meaning both sides looks exactly the same. Now, water is interesting because if you look at water molecule, right? Notice how I draw my water molecule. Like so. Right? If you draw it like this, it is not a good representation of the actual two-dimensional shape of water molecule. And the reason water molecule is like so is because if you look at oxygen, it has two pairs of electrons there. And because electrons are negatively charged, they repulse each other and they will actually push the hydrogen to the bottom, thus generating a slight negative area and slight positive charges of this generating polarity. And when you start building the molecules using the models, you can actually see the two-dimensional shape because the models are designed in such a way that when you attach bonds to it, it forces the molecule to adopt a particular shape. So always build the model before you draw it, if you can. Right? If not, on the textbooks on the side of the room, they actually have the models there. The diagrams are there. All right? Any questions? Yes. Why would a double bond or triple bond happen? You know, I don't really have a good answer for that, other than in terms of fatty acids, in terms of synthesis of fat molecules, it is just the way the organism actually synthesizes it. When you look at a plant, how does a plant actually put those molecules together, and what enzymes are available to synthesize a particular molecule? So certain vegetables, certain plants will synthesize more unsaturated fats, and certain plants, I believe peanut oil has a high concentration of saturated fat. And why do they do it? It's because the plant, when they are making the fat molecules, they actually assemble it slightly differently. In animals, in us, for instance, most of us, when we don't really make our own fat molecules. So whatever we consume, whatever we put into our body, is what we use to synthesize your phospholipids or cell membranes. What you use to make your steroid molecules is what you use to make your hormones, stuff like that. So a lot of it depends on what enzymes we have in our body that allows us to synthesize things. And in chemistry, I think the occurrences are, well, since I lost the chemistry, the occurrences of double bonds versus triple bonds. I'm not exactly sure what forces would have done that, but double bonds and triple bonds will make molecules of different three-dimensional shape, which will result in different properties, which will result in them behaving differently when involved in a chemical reaction. So it's
bonding to another, it's basically bonding to another electron. It's like an electron bonding to an electron versus the orbiting around the whole thing.
It would be more orbiting around the whole thing. It wouldn't be the electrons bonding to each other. It would be because of their attractive force and because of the way the electrons are orbiting around the two nuclei that holds them together. More so than the electrons are actually hanging on to each other. Because if you think about it, electrons are negatively charged, so they will actually repulse each other. So that is not the reason why bonds are held together. Not because of the electrons, but because of the orbit of the electrons. And of course, we talk a little bit about the forces. The forces associated with the positive and negative charges is actually quite small compared to the attraction between two nuclei. The mass of the attraction force associated with the mass of the atom. Right? But that gets a little bit too far into things that I'm not particularly comfortable with. Yeah. technical chemistry professor.
You'll know a lot more than I do.
Any questions? If not, I believe I have 1, 2, 3, 4, 5, 6. I have 6 moles of